So thank you very much for the organisers for inviting me. What I'd like to do over the next 25 or 30 minutes is really try and give you a whistle-stop tour through Exscientia's underlying philosophy, how that philosophy is physically applied in terms of how we use kind of AI, machine learning in small molecule design. For us, the critical importance and why we've made a significant investment into physical automation to allow us to not only to generate our own molecules, but to actually generate bioassay data on that. And then hopefully, kind of at the end of the talk, you'll start to see the kind of components that we think you need to put together if you even want to contemplate getting to a position where one day we might be able to actually do autonomous design. I think the challenge that exists for all of us is on this slide. And it's kind of, even today, which is like 14 years after Lilly and the Paul et al. Paper came out, people that work in applied modern drug discovery, they leave their house each day and we fail. We fail more often than we succeed. If you look at kind of current modern contemporary drug discoveries, that even today, for every 100 molecules that enters clinical trials, kind of less than 10 of them actually make it to registration. In oncology, it's even worse than that. It's about 4. If you then factor in the cost of that failure, and then so your attrition weight, the cost is that for every new drug that's launched today, the cost is at least $2.4 billion and rising. And our time horizons are still long. It's about 10 to 15 years between kind of a regional idea and actually getting a drug approved through, say, the FDA. We've got to address this, is that we live on a planet where approximately 8 billion people today. Across the globe, on average, each one of those people living to at least 73 years of age. This is just unsustainable. The rate of return today, I don't know how investors actually can make sensible investment choices into this sector. The rate of return is about somewhere between 0 and 1%. If you kind of, so collectively, we need to do something about that. And Exscientia are trying to kind of use AI to reimagine a process to fundamentally change the inputs. So this is a kind of graphical way of thinking of what kind of Paul said about 14 years ago. And there are 3 things you need to kind of really attack if you want to fundamentally change the economics of a drug discovery program. First and foremost, you've got to fail less. If for every 100 molecules you take into the clinic, only 4 to 10 make it through, you can imagine the kind of cost burden that creates. So we've got to do things about the translatability of our models. A friend of mine once joked quite recently that in cancer, every molecule that enters a clinical trial has cured cancer in a mouse, and yet very few of them actually kind of go on to be successful in humans. That tells you about how bad the translational models are. But we also need to kind of accelerate the speed and reduce the costs, so change those inputs. Because the pressures that exist today in the marketplace, the Inflation Reduction Act, loss of patent exclusivity, these are the kind of components that fundamentally drive a kind of healthcare system that's basically it's just not sustainable anymore. It's just too expensive to actually kind of to develop these drugs. I'm going to primarily talk about drug design and our capabilities in this space today. But from our perspective, there are 4 things that you kind of need to try and get right in order to address not only the problem of attrition, but also speed and cost. So clearly, I think a lot of the posters here today and some of the talks are about picking the right target, picking the right pathway. If there's fundamentally not a strong link between a target and a pathway and its cause related to disease, and we're all looking for kind of sort of novel targets, we've made some investment in this space, primarily through use of primary patient tissue in an oncology setting to try and help us not only kind of infer relationships between novel targets, but also to help to potentially pick patients that might respond to our drugs. So I'm going to focus very much on the drug design piece and why. There was a recent publication by the Boston Consulting Group in Drug Discovery Today, I think about 6 months ago. And they did an analysis of the last 10 years looking at sources of failure. And one thing that still astonishes me today is that the rate of attrition in phase 1 clinical trials is about 50%. Now that's quite interesting but bad news as well, because a phase 1 trial is not an efficacy-bearing trial. It's actually a safety. Does your compound, as it got, if it's a small molecule, is it orally bioavailable? Is it safe? Often there's a kind of PK/PD component, so does it get to its site of action? Does it modulate the target you thought? So 50% of the time, those drugs are basically, they're never even making it to efficacy studies. They don't even test the biology. And so I think that tells us that even today, one of the common issues we have to address is actually molecular design. Because the thing with small molecule design, actually, but any modality actually, but particularly small molecules, is that the properties of a drug are cast on the day you draw it on a computer screen or a piece of paper. You can slightly modify solubility characteristics through formulation, but the way that drug's going to interact with the human body, the way that drug is going to interact with kind of a protein network is fixed at the time you draw it. So design is very, very important. Just a little bit of just one shout out to Exscientia. So Exscientia's been around for a long time before AI became the fashion and the hype that it is today. It started out as a spin-out from the University of Dundee back in 2012. And grew really slowly, kind of grew in the real world, not sort of the big investments that you see today. And over the last 12 years, we're proud of several things. We've actually managed to get 8 development candidates, both internally and through partnerships, into clinical development. We run a dual business model where we both work in partnerships with academics and with large tier 1 pharma, but we also have our own pipeline. Even in the absence of kind of some of the automation I'm going to show you later, we've been able to kind of dramatically change some of the inputs already. So we've been able to show that basically the approach I'm going to walk you through over the next few slides, we're seeing about a 60% reduction in the time it takes for us to actually kind of build an assay cascade and get to a molecule that looks like it has the promise to be a development candidate. We're also very efficient in the experiments we do. We are a compute, but an experimental science organization. We use the kind of benefits of active learning to really understand which experiments will actually generate the most data in the most efficient way. So we make far fewer compounds than a typical modern drug discovery program. We generate kind of, by doing that, we're actually testing fewer compounds as well, and that directly translates in today's marketplace into about an 80% improvement in capital efficiency. So the kind of abstract for this talked about humans. We are an expert-led organization. Yes, we are striving to try and get to a place of autonomous design, but this is one of the places where kind of human scientists are really, really important. It's actually defining the problem, either defining the target candidate profile, really understanding what does the patient need? How might your drug be used? Is it going to be used alone? Is it going to be used in combination? Is there any existing data? And here's just a section of some things that we've kind of spoken about publicly. So whether it be from PKC theta, where around 15 to 20 companies tried and failed to actually get a selective small molecule into the clinic. So this is a target that sits on the immunological synapse. Through to LSD1, where we looked at the kind of patients that we're likely to treat with that mechanism. One of the potential utilities of LSD1 is in small cell lung cancer. If you look at what happens to those patients when they first present, about 25% of them already have brain metastases. And during the lifetime of that disease, more than half of them will develop brain metastases. And more often than not, it's the brain mets that kill the patient, not the primary. So we deliberately designed a molecule that was brain penetrant. LSD1 is also important in you and I. It's kind of one of its functions is in the maturation of certain blood cell types, such as platelets. So there's a therapeutic index argument here as well, is that you can't just kind of irreversibly or permanently inhibit that enzyme because you will deplete platelets. And so we designed a molecule that's brain penetrant, was reversible by mechanism, and has a proposed short half-life in humans so that you're on target to do to gain efficacy, but you're off target long enough for that enzyme to recover. And so these are the kind of, before we even start designing molecules, we spend a lot of time writing down what the target candidate profile is, because that then sets the reward function by which we design against. A couple of philosophical slides, but they're really important. We believe that novel drug design is actually a learning problem, and that you can't just compute your way to a solution. That might sound a little bit strange coming from someone that basically works at an AI company. But here's the problem. So hopefully most of you are aware of kind of Chris Lipinski and his team at Pfizer back in the late 90s. Chris and his team, I think it's still a useful kind of rule of thumb, define the kind of sort of baseline properties that it takes for a small molecule to be bioavailable, to basically stand a fighting chance of being a drug. Well, if you enumerate, if you take carbon, hydrogen, nitrogen, oxygen, the atoms that basically make up small molecules, and say, okay, what can I make? What won't explode? What's stable? And then fundamentally overlay that with kind of, we call it Lipinski space. There's some big error bars in there, but it's somewhere between 10 to the power 30 and 10 to the power 60. That's your search space. To put that into context, I think modern astronomers think the number of stars in the known universe is only 10 to the 24. So it's bigger than that. If you think each one of those stars, if you like, is a potential drug, is that if you're going to compute your way to an answer, you need to predict about 2,500 properties. A drug has to be, it has to get through the GI tract, it has to get absorbed, it has to get to its kind of target tissue. It has to not hit a lot of other targets as well. It has to not interfere with sort of basic metabolic systems as well. And that there are only basically a few solutions for any one of these problems. It's also a sparse data problem. Granted, I think big data in the sense of kind of property predictions, sure. So being able to predict kind of ab initio how soluble a novel compound might be is largely lends itself to kind of sort of big data and to those predictive models. But by the very definition, if you're working in a novel kind of target space, that there is no information. You can perhaps predict maybe the kind of protein structure. So again, thinking of good examples, or Kemble, which some of our Accenture employees helped to kind of found, is that even Kemble today, I think in its latest version, has about 2.4 million actual molecules that have been hand-curated. It's about 20 million data points. So even though that's a good source of information to build models from, and I think we and others do use that, it is like me giving you a bucket, you going to the North Sea, taking a sample of the North Sea and go, I've just sampled the world's oceans. No, you haven't. You really haven't. And that at novel targets, there's little to zero data. So you have to create a system that is capable of learning its way into a problem. That's our central philosophy. Who's old enough to remember 20 questions? Who's young enough to have played Guess Who with their children? So this is a little interlude about information theory and why learning your way into a problem always beats screening. So there are around about 600,000 word forms in the English language. Only about, granted, maybe 30,000 actually in common use. Now, if I was to ask one of you in the audience, and I won't pick anybody out, but to look, I'm going to pick a word from that 600,000. Would you rather have 20,000 guesses, or would you rather have 20 questions with a guess at the end? This is what information theory tells us, is that if you were just to kind of make a random guess, is that you will fail just less than 97% of the time. Just simple probability. Information theory tells us something a little bit, this is why questions are important, is that if you can ask a perfect question, and a perfect question divides a data set in half. So if the people have played Guess Who, it's like, does the character have blonde hair? And you basically kind of close those. We're asking a question in kind of drug discovery. So if you ask 10 questions, even at the end of that 10 questions, you've really only still covered 0.1% of that space. You get to question 15, you've covered a bit more, but there's still 97% of that space that's explored. However, when you've asked 20 perfect questions, you should get the word right because you divided the data basically so finely. And this is how we think about how humans learn as well. It's about iterative learning. Is that a few interesting things come out of this, and they really matter, is that if you look at that, is that most of your learning is done at the end. The last 5 questions are really, the first 15 are important, but the last 5 are basically what gets you across the line. So if you replace 600,000 with 10 to the power of 60, and for us, if you replace a question with, we're going to physically actually make and test a drug that we've predicted should work, that's the kind of underlying philosophy. So for us, what matters here then is like, okay, how fast can we get answers to our questions? And how can we ask the most efficient questions possible? So does this sound familiar? Because it should. It's how humans have perhaps done some of their finest work. It's about creating near-term prototypes, iterative learning, feeding information back into that system, doing it as efficiently as possible. You can apply it to the Mercury, Gemini, Apollo missions. It was like little steps at a time. Kind of try not to blow up on the launch pad too often, but learn your way into a problem. So predict and design is where AI is our ally. It that allows us to kind of ask efficient questions. Make and test. We need to kind of understand whether the compounds we've produced and predicted are actually any use or not, and how close to predictions were they. And it's about how fast can we basically drive that design, make, test loop. So what does iterative learning look like in practice? So again, this is just a data slice. This is the first, so those 8 compounds. So I went back and looked in our system and said, how many novel compounds did we make during the entire lifetime of the project? So not just the one series that led to the candidate. The initial exploration of the chemical space, the backups that we all want, the patent examples that we need to produce. And it's typically about 400. So it's about 80% less than the couple of thousand I would have made when I was at Evotec maybe, or even at Merck. That gives you a sense of like the kind of efficiency in terms of what the kind of questions we're asking. Why does that matter financially? Even if you're using a contract research organization that's maybe charging you, I don't know, $100,000 per FTE, I reckon that the cost to synthesize one compound and to get an initial data set that's useful is somewhere in the region of $10,000 per compound. And it's only going one way with inflation. So you've got to be smart about the kind of number of compounds that you make and test. And of course, I'll come on to later about the automation studio is that we have to be wise to kind of sort of geopolitical issues around the world and the risks and the cost arbitrage that come with working with Asian CROs. Busy slide, so I'll spend a couple of minutes on this. We've built, because of that philosophy, we've built our design platform, which is called Centaur, to address a lot of things. So first of all, it has to be comfortable with a variety of data sources. It has to be able to design from not just a protein structure. Has anybody here who's doing phenotypic screening, where you don't know what the underlying mechanism is? Anybody? There's a few of you. How is AlphaFold going to help you in that environment? It's not. If you don't know what the structure is. So it just gives you a sense that you must be able to use any kind of data set, whether we've used video tracking from animals, we've used transcriptomic kind of changes when you perturb a cell. So you get the point, the data can be anywhere. It can be from the literature, it can be from Chembl, it can be data we've generated, data our partners have given us. It could be crystal structures, it could be predictor structures. You can't be wedded to one method either after that. So you think about the variety of kind of ways you can analyze and build models, whether it be sort of Bayesian optimization or deep learning, simple transformers, is that I think the message to take from this slide is that we're systems integrators. We spend a lot of time and money making sure that our kind of models are predictive and effective. But we're not so proud that we basically won't leverage basically what's free and in the public domain, but we integrate them into kind of a workflow. And the way the design platform works, and I'll show you kind of a video schematic, is that ultimately it's integrating those data sources. We set a reward function. It's literally written in Python script. So we take a written target candidate profile. What do we want the molecule to be able to do at the end of the journey? And we encode that into a reward function. The system on each generative cycle will kind of search probably 1 million compounds or so of virtual space. And will produce a kind of set of molecules that it believes actually meets the kind of the parameters for that design cycle. And then by a process of active learning, which I'll describe in a second, we then select the smallest number of compounds that generate the most data. Just two things before I show the video. So active learning. It's actually many things, but in the sense of, the best way to explain it in a small molecule setting is that, to pose the question, why would you make 100 compounds when the data from 10 would give you the answer? But how do you do that? So what active learning allows you to do is to use very rigorous mathematics to look at a data set and a question set. So, okay, it's a bit like the 20 questions. Which of these compounds should I make to most efficiently not only advance the project, but fundamentally improve the underlying predictive models that are being used for that? The merit function is a little bit more familiar for people that work in this space. So I think you're all familiar with like multi-parameter optimization. Merit is just a version of that. We typically use maybe about 8 different functions. It could be early in the program, it could be a combination of how well are we predicting solubility, on target, biochemical activity, cellular activity, for example. With a twist, it's a geometric function for us. And that matters because if you're familiar with how geometric functions work, if one of those parameters tends to 0, the whole function tends to 0. It forces us to basically design balanced compounds. You can't get away with high potency and kind of god-awful solubility, for example. So this is a kind of schematic of how kind of one of our generative systems works. It's called Gambit. It tends to work really well when you've kind of locked in on, say, a novel call that you want to fix, and then you want to enumerate that call using not only kind of synthesis-aware design. You can use it in a kind of 2D kind of pharmacophore one. This is actually kind of docked back into the protein. And it's kind of the best way we try to figure out to show an audience basically kind of what generative design looks like and how the compound is trying to virtually score compounds as you go along. So what you see here is that the kind of system's enumerating compounds on a fixed score. It's predicting, in this case, properties like potency, selectivity. Sometimes we put in a more advanced form of the project or a later form, human dose prediction. And it's kind of searching through that space and kind of predicting kind of overall properties. And from this, what we produce is then this kind of the smaller subset of compounds that we subsequently take into actually physically making and physically testing to know how well our predictive models work. One of the kind of historical issues, actually, with generative design is like, it produces compounds that are just unbelievably difficult to make. For humans, never mind kind of automation systems. And so that was kind of why Accenture in its early days kind of hired medicinal chemists, kind of practicing synthetic organic chemists to understand kind of and how to design and optimize a system. We've gone one step further than that over recent years, kind of both in collaboration on our own, generating a system that, think about, if you're going to make a compound, you've got to be aware of so many things. Are the building blocks actually commercially available? There's no point designing a molecule and waiting 12 months for a raw material to turn up. You want to try and get your synthesis right first time, because every time, I think a lot of money is actually wasted in drug discovery because the trial and error that still exists today in kind of contemporary synthesis. There are lots of tools out there, people that use SciFinder or Reactis. If you want to draw a novel structure, there are modern kind of tools that will give you a good sense of whether that compound is synthesizable or not. The problem that we face is like, yeah, but we want to do that at 10,000 molecules. We want to be able to kind of know how difficult or easy something is before we actually go ahead and make it, and then factor that into kind of the timelines of the project. So I'm going to run a video here. This is actually a real screenshot. So this is the platform. So we've kind of run a generative design cycle. Active learning's picked about 15 compounds. The human here is basically going to go back into the system, ask a question. Basically, were any molecules made when nitrogen was at this particular position? It's then going to look, oh, how did they dock into the kind of protein in this particular case? And it will finish off by looking at kind of synthetic kind of plausibility and kind of root outcome. So we see here is basically that histogram gives us a sense of like 10 to 12 days is what the system would think it's going to take 50% probability to actually make the molecule. The human's gone back in, looked at how well that kind of compound docks, and then this is an interesting screen that you can actually then look through the synthetic route. It gives you a sense of real lead times of the kind of reagents. You can change the synthesis around and swap them out if it's basically, it might be cheaper to actually make a building block or quicker than to basically wait for the lead time. That's really important. We're already seeing benefits in terms of the productivity at the CROs we use. They're already 30 to 40% more efficient after the use of this. But it had to be addressed if we start to try and think about how can we make bespoke synthesis automation efficient. So for just the last few slides and the last part of my talk, I want to talk about why we made a $50 million investment into an automation facility. If you think about that learning loop before, you design a compound, you make a few of them, you test them, and you learn. The make and test has always been the bottleneck in human drug discovery, whether it's humans making compounds or even robots. But we reached a point where our kind of operational models was we would design compounds, our contract research partners or our partners would make the compounds. Over the last few years, we'd created a capability to test them, but we'd really pushed the limits of that operation, so we needed to try and figure out a way to kind of bring that back in-house and to try and accelerate our ability to learn. And so at Milton Park, kind of just south of Oxford, we took a building about 3 years ago, and the building is part of the architecture. It's about 2,500 square meters with kind of a state-of-the-art kind of closed-loop robotic system in there. This is what it looks like in practice, is that essentially it's a square. It starts off with kind of a reaction station that's kind of orchestrated by our software team. We actually analyze the compounds, we purify them, we don't use crudes. We use kind of predictive NMR techniques to make sure understand are we actually making what we thought we'd made. The compounds are generated into acid-ready plates, and then kind of biochemical and cellular data is kind of generated, and that then feeds back into our underlying system to update the kind of models. One of the things a challenge I'll leave you with is that I'm old enough to remember like the first wave of solid phase synthesis, where the world believed that if we just made enough compounds kind of on polymers, is that one of those would be a drug, which is clearly fantasy. But one of the benefits of that generation is that a lot of the chemistry equipment that you might use today, particularly kind of automated purification, that came from that generation. But even today, as we've kind of built our closed-loop system, there's so many engineering challenges that we still need to overcome. There's the kind of incusites you use, it could be the chemistry workstations. They were meant to interact with fingers and thumbs. They were not meant to interact with automation. And so we've got to figure out ways of how do you bridge gaps where there's no engineering solution. And so there's a few, these are just using other people's systems. So this magnetic levitation system that I think comes from Zinsa allows us to kind of transport kind of fractions from an HPLC or an SFC system basically to a Genovac. We go through a lot of consumables such as solid phase extraction tubes, and so kind of the mobile cobots that kind of map out the space can kind of solve that. And our friends at Hi-Res basically with their overhead conveyor system allow us to basically create more space around, basically, so we transport plates above head height rather than from a kind of single robotic workstation. We're already seeing some of the kind of efficiencies here is that it's not just on the kind of chemistry side, but the way that even our biology system is a learning environment. To give you an example is that humans come back into this again. We have the human scientists parameterize the assays that we're trying to build. So, okay, what are the variables that we want the system to look at? But then we walk away and let a design of experiment system run to basically optimize that particular assay. The system's then configured so that if a similar assay comes along another week later, it's already got a starting point. So to give you an idea is that kinases are still really popular in modern drug discovery. Now, you can send your compounds out to kind of sort of the biggest CROs to get panel testing, but we wanted to kind of generate data in-house to build models from. So we looked at the kind of kinome, kind of from a structural bioinformatics perspective, said, okay, if we kind of make assays to these 80, that will give us pretty good coverage of the kinome. So then we asked the person to basically, using the automation platform and design of experiments, can you develop those assays? It took one person 2.5 months to develop 75 assays. If anybody basically works in a laboratory environment, that's some going. Robotics also allows you to do things that you'd expect, but it's still good when you can see them happening. So this actually runs 24/7. You can basically eat your cornflakes in the morning and basically look and see how the kind of assay runs are going. And we're already starting to see, basically, we've been making compounds on this system for a couple of months now. And I'm just about done. That's my last slide, is that hopefully, I haven't given you any specific cases, but hopefully we've got a sense of how we're using artificial intelligence. It's not black box. It's mathematics is probably harder than Cambridge Part 3 mathematics, but it's not black box. Hopefully you get a sense that how we truly believe is that what we're trying to do is that to use the benefit of computational systems. Humans are not very good at multi-parameter optimization because there are just too many variables involved. But by using kind of how humans themselves iteratively learn and just applying computational techniques to multi-dimensional problems, that's kind of what we're trying to do here. Hopefully you got a sense of like why we've made the journey into physical automation and why we've had to orchestrate that kind of just running a model where we design and then somebody else makes and tests, kind of just pushing our limits. And that hopefully you've got a sense that back from the title is that the components here may one day allow us, in terms of using decision capture, to even drive an autonomous design system. And with that, I'll stop and take questions.
Thank you, Dave, for that glimpse of the lab of the future. We've got time potentially for a couple of questions, and there's some roving mics, so there's one at the front there. Where's Mike's just coming down the steps.
Thank you, Dave. Dave, what about the learnings from, say, with your AI design workflow? You've got the molecule, fine, but then when you're doing a clinical trial, and there's a lot of data where clinical trials have failed, but you've got the structure of the molecule, you've got the data in human, are you able to overlay that as well, potentially, or would you look to overlay that in a future version of this workflow where you've got real translational data? So that's one question. Second, quickly, there's a generation of us who remember combinatorial chemistry and are scarred by combinatorial chemistry. The thing with AI, are there learnings from the combinatorial chemistry that a company like Accenture, who have been at the forefront of AI drug discovery, are putting into practice? Because, as we all know, with combinatorial chemistry, you got to the point where most drug discovery companies, all they cared about was how many screens they did and how many crap compounds they started with, and obviously we know what came out. So how is that working with Accenture?
I'll take your first question. So the answer is yes. So in a narrow area where we focus our own investment dollars, so that's in oncology, we acquired a company that what that allows us to do is that they can take primary patient tissue, either from kind of hematological cancers, which is kind of where they start their journey, or solid tissue. And we use that in a variety of ways. One, if we're kind of, say, taking a functional genomics output, but okay, what does that target look like in a 50 sample population of AML samples? Because what you need to do is, this comes back to the translational model, is that the translational models, whether it be in psychiatry or oncology, need to be better. A joke, but it's not a joke. I think we've cured cancer in mice so many times and then not been able to translate that into humans. So we take that primary human tissue, we can test compounds on it. We can also help to try and select patients. So we're embracing, the problem again with rodent models is that they're all genetically pure. They're all identical, is that humans are not identical. So we try and capture the heterogeneity. It's fine to say, okay, we'll test 10 samples, 6 really respond well to the drug, but 4 don't. Because then you can ask another question, well, what's the difference between those samples that say a genetic or a transcriptional level? So then, so we apply that in terms of, to some degree design, but more into kind of sort of target and patient selection. Your second question, I think what we've learned, and it's kind of that DNA encoded libraries is kind of part of that kind of combinatorial chemistry. I think those methodologies allow you to try and get a chemical foothold into, say, a new chemical space. But we can't kid ourselves. I think the search space is so large, is that we truly believe that you can't just use AI to get your way to a drug. You've got to be able to kind of generate in a very tight loop data that tells you how good were your predictive models, make your predictive models better, and then that learning your way to a solution is much more efficient than trying to screen your way.
There's last question to Davide, up there.
Thank you. Just a quick follow-up on this. I believe you refer to all-site acquisition with pharmacoscopy. So is the scope of this to refine the clinical trial piece, or did you actually already manage to bring this up to a sort of discovery phase where the lessons learned from the sort of stratification can be used to kind of build drugs that are already targeting a specific subpopulation?
Yeah, so we're using it, so just quickly for the audience, is that Allsight was a company we acquired, is that they still are the only company I'm aware of. This was in a heme-onc setting where patients that had no standard of care left, so these were 4th, 5th, 6th line treatments, largely Austria, southern Germany, their biopsies, so lymphomas and leukemias, were physically sent to Allsight premises, and using a proprietary system over the course of about 5 days, a drug list of about 140 drugs, including sort of biomolecules, basically was evaluated. That particular endpoint was a very simple one, kind of relative blast fraction. But we're able to basically then go back to their physician and say, okay, we think this particular patient would respond best to this drug. And then the physician took that data point, either chose to do what they wanted to do or basically to took our data point. This is all published, and basically what we saw was that, yeah, probably half the patients in both arms, because they were so poorly, unfortunately, they passed away within 12 months. But about 30% in our control arm basically went on for 2 years or more with progression-free survival. So that gave us a sense of that there's some clinical validity there. To come back to your specific point, yeah, we're using it on LSD1 at the moment. So the first entry point to the clinic will be in AML. So that's allowing us to kind of look at kind of what mutational status, perhaps at a genetic level, kind of impacts whether kind of super responders from poor responders. But we're also using it in a discovery setting as a part of our target discovery effort is about taking annotated compound sets and trying to triangulate, well, what is actually the common understanding, because like when you've got, say, a load of, say, tyrosine kinase inhibitors, and some of them work and some of them don't, yeah, but what's the common pathway? And that's allowing us to kind of uncover sort of novel potential targets as well in that space. So we are using it in anger both for patient stratification and for basic discovery.
Fantastic. Thank you, Dave.